A very good morning to all my dear students. Let's go through the industrial pharmacy 2. So, dear students, we are on to the WHO guidelines for technology transfer in case of pharmaceutical manufacturing. Now, it became very clear for we people that what is the technology transfer. That means the shifting a particular technology from one place to that of the another place. It might be intra-transfer, it might be inter-transfer within the two different sites. And what are the guidelines that are published by the WHO as far as these transfer processes are concerned? We are going through that one. While studying these WHO guidelines, from understanding and for study point of view, these has been categorized into the different headings like say introductory guidelines, scope, related guideline, glossary, that is the terms that are used during the technology transfer, the guidelines associated with the organization and management, then the guideline associated with the production, transfer process, packaging and cleaning, quality control, analytical method transfer guidelines, premises and equipment related guideline, documentation related guideline, and guideline for the qualification and the validation. As far as all these 8 to 9 different subheadings are concerned, we had already finished the first 4 and we are on to the production, transfer, processing, packaging and the cleaning guidelines. That already we have started in our previous lecture, in which around 5.7 up to 5.7 we had finished, we had covered, we had gone through these guidelines in our last lecture. Now, in continuation to that, today we will start with the 5.8, that is the guideline associated with the production transfer, which are in relation with that of the processing, packaging, and the cleaning. So, what are the 5.8 guideline that will tells us? The 5.8 guideline that will explains that the SU is what? Sending unit. The SU should provide a detailed characterization of the product, including its qualitative and quantitative composition, physical description, method of manufacture, in-process controls, control method, and specifications, packaging components and configurations, and any safety and handling considerations. So these guidelines are related with that of the product, related with that of its quality, related with that of its quantity, related with that of its description, the method of manufacture, the process control, the methods of control, specifications, the packaging related components, the configuration and safety as well as handling of the product. That is the 5.8. The 5.9, it will tells us that the sending unit should provide any information on the history of the process development which may be required to enable the receiving unit to perform any further development and or process optimization after successful transfer. Let us say for example, for during the discovery of antibiotic penicillin, it is the serendipity. So this information should be provided by the sending unit, or the technology receiving unit should know this particular history. So based on that, they should come to know that how the antibiotic penicillin that had developed, or how the penicillin antibiotic that came. Such information may include the following. Now, what the history related information is required? So that is in detail has been given. Information on clinical development, for example, information on the rationale for the synthesis, route and form selection, technology selection, equipment, clinical tests, and product composition. This information needed to be provided by the sending unit to that of the receiving unit. In addition to that, the information on scale-up activities, process optimization, statistical optimization of critical process parameters, critical quality attributes, pilot report, and or information on pilot plant development activities indicating the number and disposition of batches manufactured. This information to be given. Then the information on report on full-scale development activities indicating the number and deposition of batches manufactured, and deviation and change control, sometimes referred to as change management. The reports which lead to the current manufacturing processes, they need to be provided. The change history and reasons. If we are doing any changes, then why the changes has been carried out, they should be reported. For example, a change control log indicating any changes to the process or primary packaging or analytical methods as a part of process optimization or improvement, they need to be provided. And information on investigations of products and the outcomes of the investigation. The guideline 5.10, it is in association with the sending unit should provide to the receiving unit information on any health, safety and environmental issues associated with the manufacturing processes to be transferred and the implications. For example, the need for gowning or protective clothing. Right. So let us say, for example, if there are any health hazards, in that case, there is need to go for wearing the personal protective equipment, PPE, isn't it? Nowadays, that everybody knows that what PPEs are, right from including the mask hood, right from hand gloves, right from the gowns, right from the shoes, coverings, etc. So, in order to safeguard the life of an individual who are handling all these things, and if any byproducts are there, what is their implication, what is their impact, what is their effect onto the environment, that also need to be discussed and provided. The 5.11 tells that the sending unit should provide to the receiving unit information on current processing and testing, including but not limited to what? But not limited to a detailed description of facility, requirement and equipment, information on starting materials, applicable MSDS. What is MSDS? Material Safety Data Sheet. MSDS is M for material, S for safety, D for data, S for safety. Material Safety Data Sheets. Yes, last S stands for the sheets. And storage requirements for raw materials and finished products. The description of manufacturing steps, narrative and process maps or flow charts and or master batch records, including qualification of in-processing hold times and conditions, order and method of raw material addition and bulk transfer between processing steps. A description of analytical method, identification and justification of control strategy. For example, identification of critical performance aspects for specific dosage forms, identification of process control points, product quality attributes, and qualification of critical processing parameter ranges, the statistical process controls SPC. Then design space in case where this has been defined. Validation information, for example, validation plans and reports related with validation. The annual product quality reviews, stability information, an authorized set of protocols and work instructions for manufacturing and environmental conditions or any special requirement needed for the facility or equipment depending on the nature of the product to be transferred. So all these are the guidelines associated with the production transfer in 5.11. What the 5.12 will tells, the guideline 5.12 that is related with, or it explains that, it tells that during the transfer process, the receiving unit should identify any differences in facilities, systems, and capabilities and communicate with the sending unit about these differences to understand the potential impact on ability to run the process to deliver good product quality. The ultimate aim of all these processes is the getting the product quality product, or the good quality product, or the good product quality, right? So during the transfer, if any differences has been happened, if has been any differences has been observed, has been found in terms of facility, system, capabilities and communication is concerned, so that should be properly communicated to the sending unit and to be discussed that what is the impact of these differences. If there is a major impact, first of all, there should not be any difference. And if any difference is there, what is its impact? If it is not having any kind of significant changes into the quality of the product, then need not to worry. But if it will affect it, then that would be taken as a significant difference, as a significant change. The differences should be understood and satisfactorily addressed to assure equivalent product quality. Yes, we should get the equal product quality based on the information received from the sending unit, and the receiving unit should consider its own capability to manufacture and pack the product to the required standards and should develop relevant plant operating procedure and documentation before the start of the production. The process development at the receiving unit should address the following tasks. The first one, comparison and assessment of suitability and qualification of facilities and requirements as far as equipments are concerned. The description of manufacturing process and flow of personnel and of material at the receiving unit. It should be narrative and or process maps or flow charts that should be provided. Determination of the critical steps in manufacture, including hold times, endpoints, sampling points, and the sampling techniques. Writing and approval of the standard operating procedures for all production operations, right from example, dispensing, granulation, blending, or encapsulation, solution preparation, tablet compression, tablet coating, liquid filling, primary, secondary packaging, and in-process quality control in association with packaging, cleaning, testing, and the storage. Evaluation of the stability information with generation of site-specific stability data if required, and compliance with regulatory requirement for any changes made in terms of the batch size is concerned. The 5.13 13 onward, these are the guidelines in association with the packaging of the transfer. So whatever the guideline we saw up to 5.12, that was related with the processing. Now the guideline that is in association with the packaging. So the 5.13, it will tell us the transfer of packaging operations should follow the same procedural pattern as those of the production transfer. Information on, that is the production transfer related and the packaging transfer related, they should be same. The 5.14, it will tell us that the information on packaging to be transferred from the sending unit and the receiving unit includes specifications for a suitable container or closure system. The same which is to be followed by the sending unit should be adopted by the receiving unit, as well as any relevant additional information on design, packaging, processing and labeling requirement and tamper evident and anti-counterfeiting measures needed for qualification of packaging component at the receiving unit. For quality control testing of packaging component, specifications should be provided for drawing artwork material, for example, glass, card, and the fiberboard. These are in association with the 5.15. 5.16, these are based on the information provided. The receiving unit should perform a suitability study for initial qualification on the packaging components. Packaging is considered suitable if it provides adequate protection in terms of preventing degradation of the medicinal product due to environmental influences. The safety in terms of absence of undesirable substances released into the product. The compatibility in terms of absence of interaction possibly affecting the medicine quality. And performance means the functionality in terms of the drug delivery. The 5.17 onward, these are guidelines in association with the cleaning process in case of the production transfer. So the 5.17, that is as far as the cleaning is concerned, what are the guidelines will tell us? The guideline tells that during the manufacturing process, pharmaceutical product and APIs can be contaminated by other pharmaceutical products, or APIs if the plant is processing different products. To minimize the risk of contamination and cross-contamination, operator exposure, environmental effect, adequate cleaning procedures are essential. So the 5.17, that is cleaning in association with that of the process of avoiding the contamination. Intercontamination, intra-contamination. Inter-API contamination, intra-API and excipient contamination, intra-excipient contamination. So these may lead to production of the risk. So that's why these should be avoided. So 5.18, that is cleaning procedures and the relevant validation are the site specific. That means for different places, for different site, these are very specific one. For packaging, it might be some different. For production, it might be some different. For quarantine, it might be some different. For reception, it might be some different. In order for the receiving unit to define its cleaning strategy, the sending unit should provide information on cleaning at the sending unit to minimize cross-contamination due to residues from previous manufacturing steps. Right? Because this is most important. Earlier we might be manufacturing something and later we might be manufacturing something. Right? So this may get contaminated and that may ultimately lead to the failing of the batch, and sometimes this may lead to recalling the batch after getting the results from the market. There is a history which will tell us that some contamination of the female sex hormones with that of the some of the product that might have lead to the development of the disorders like say the gynecomastia and all. So that totally cause to recall the whole batch from the market. So the contamination that is of great significance and that should not occur. So The information on solubility of active ingredients, excipients, and vehicles, minimum therapeutic doses of the active ingredients, the therapeutic category and toxicological assessment, and existing cleaning procedures. Some additional information that is needed to be provided as appropriate and wherever applicable. For example, this is in association with the cleaning validation reports in terms of chemical and microbiological contamination, information on cleaning agents used, efficacy, evidence that they do not interfere with the analytical testing for residues of APIs, removal of residual cleaning agent, and the recovery studies to validate the sampling methodology. The 5.19, it will tell us that before the transfer, the sending unit should provide information on limits for product residues and the rationale for limit selection. 5.20, it will tell us that the based on information provided by the sending unit, the cleaning procedure should be designed at the receiving unit, taking into account relevant characteristics of the starting material. Now, these characteristics might be, for example, the potency, toxicity, solubility, corrosiveness, and temperature sensitivity. The manufacturing equipment design and configuration, cleaning agent, and product residues. The 5.21, these are related with the implementation of all these three different steps, that is processing, packaging, and cleaning. The implementation of processing, packaging, and cleaning system. The 5.21 onward, these are in association with this, and what it tells, it will tell us that the trial batches, that is the demonstration batches, that are normally provided to confirm process capability before initiating formal validation, where trial batches are produced at a minimum. All clinical processing parameters and finished product specifications should be assessed. There is a need to produce the trial batches, and that too in a minimum number, and at those minimum number they should meet the all finished specifications, and that should be assessed and that should be confirmed. The 5.22, it will tell us that once the process capability has been established at the receiving unit, assuring that the product, process, and method at the receiving unit meets the predefined and justified specifications. Process validation and cleaning validation can be carried out. What are the result, what are the specification that was made by the sending unit, the same kind of specification, the same kind of result that should be achieved, that should be received by the, that should be obtained, that should meet by the receiving unit. That is the ultimate outcome of that guideline. Now, after fifth guideline, that is the production transfer related with the process, packaging, and the cleaning. There are guidelines prescribed by the WHO which are in association, which are related with the quality control. So, in case of any product, in case of any manufacturing, more specifically in case of the pharma manufacturing, there is a great concern of the quality, and there should not be any compromise to that of the quality. So there are guidelines which have been given by the WHO, and these guidelines should followed by the manufacturing units. Now what these guidelines will tell us, so the guideline that will start from the 6.1. So 6.1, it will tell us that the transfer of analytical methods should accommodate all the analytical testing required to demonstrate compliance of the product to be transferred with the registered specification. Analytical methods used to test pharmaceutical products, starting materials, packaging components, cleaning samples, if applicable, should be implemented at the testing laboratory before testing of samples for process validation studies is performed by the receiving unit. The process validation samples may be tested at the receiving unit and the sending unit or the third laboratory. In order to get the unbiased results, there is need to confirm the testing at receiving unit, sending unit, or sometime by the third party. And these are in terms of, these are in relation with that of the product, starting material, that is said to be the raw material, the packaging materials, that is say the glass containers, PVP containers, or cardboard containers, cartons, and the cleaning sample, the material which are used in order to clean the plants. The 6.3 QC guideline, it is said in it is in association with a protocol defining the steps should be prepared for transfer of analytical methods. The analytical methods transfer protocol should include a description of the objective, scope, and responsibilities of the sending unit and the receiving unit. A specification of materials and methods, the experimental design and acceptance criteria, documentation, including information to be supplied with the results and report forms to be used if any. Procedures for the handling of deviations, references, signed approvals, and details of reference samples. That is starting material, intermediate, and the finished products. So it is during the analytical method transfer. It is the implementation of the method which are used same as that of the sending unit by that of the receiving unit. The 6.4, it tells that the sending unit's responsibilities for the transfer of analytical methods are, now what are the sending unit's responsibilities in terms of the QC guideline? These are, they provide method specific training for analysts and other quality control staff if required. They should assist in analysis of QC testing results. They should define all methods to be transferred for testing a given product, starting material, or cleaning samples. They define the experimental design, sampling method, and acceptance criteria. They provide any validation reports for methods under transfer and demonstrate their robustness. They provide details of the equipment used as a necessary part of validation report if available and any standard reference samples. They provide approved procedures used in testing and review the approved transfer reports. So all these are the responsibilities of whom? So all these are the responsibilities of sending unit as far as QC guidelines are concerned, and these are in accordance with the 6.4. So 6.4, that will explains what? The 6.4 that will explain the sending unit's responsibilities. Now followed by sending unit responsibility, there are certain responsibilities which are associated with the receiving unit, that is 6.5. So what are the responsibilities of the receiving unit? These are to review the analytical methods provided by the sending unit and formally agree on acceptance criteria before execution of the transfer protocol. So whatever the method that has been provided by the sending unit, as such, blindly they should not be adopted by the receiving unit. They should review all these processes and they should give agreement of acceptance of all these criteria of all these methods. Next, they should ensure that the necessary equipment for the QC is available and qualified at the receiving unit site. Whatever the equipment that are mentioned, whatever the instruments that are mentioned, they should be fulfilled by the receiving unit. That is the duty, that is the responsibility of the receiving unit. The equipment used by the receiving unit during the analytical transfer should meet appropriate specifications to ensure the requirement of method or specification that are made. They should ensure that adequately trained and experienced personnel are in place for analytical testing. The trained and experienced personnel, that is the manpower requirement, that is the duty and responsibility of the receiving unit. They should provide a documentation support capable of recording receipt and testing of samples to the required specification using approved test methods and of reporting, recording and collecting data and designation of status, that is approved, rejected, quarantine. Likewise, they should execute the transfer protocol. They perform the appropriate level of validation to support the implementation of the methods, and they should generate the obtain approval of transfer reports. So all these are the responsibilities in association with that of the receiving unit. Their duty is to ensure that everything in accordance with that of the requirement for the QC department, that is equipment, manpower, then instrument that should be provided, and all these steps that are in accordance with these standard specifications.